Okay, so, did I press it? Yep. Yeah, I'm going to be here. There's still some career sessions and everything tomorrow, but, pardon? No, I think that's Friday. Friday's in this room, but tomorrow morning you have more sessions. I don't know if there's anything in the afternoon or not. Okay, so we only have like 10 minutes, 15 minutes, so if you could take out your booklet on cloning. We spent the majority of the class going through the cloning booklets, so hopefully having gone through that activity, you have kind of a good idea of what is meant by cloning and a lot of the pros and cons, and we've covered most of the chapter objectives for it. Okay, so why would we talk about cloning when we're talking about mitosis? Yeah, because the cells are caused to undergo mitosis in order to form an organism or to develop tissue or whatever the case they're using the type of cloning for. Okay, so there are different types of cloning. The activity you guys did online focused mainly on reproductive cloning. Okay, but there are other types. So we're going to look at the three main types so you can understand.
Ben,
Yes?
Corey from 3D Datacom is on the phone regarding the smartboard in your room not working.
Can I call him back in about 10 minutes? Okay, thank you. It's not mine. Okay. So there are three main types depending on what cloning is used for, the purposes. So if it's reproductive cloning, what is the end result, do you think? Taylor and Matthew. You've had most of the class away from us, so please pay attention to this last part. Okay, what is the end result of reproductive cloning, do you think? Yep, an organism, usually. Okay, so you'll form a zygote, an embryo, make that embryo develop, start dividing, put it into a surrogate, usually, and it will develop into an organism. Yep, a clone of another thing. Therapeutic cloning is another type we're going to look at. And the result is not an organism, but it's another type of cloning that's important for a lot of the research they're doing these days. You guys want the lights out a bit? Okay. Enough? Okay. Okay. So reproductive cloning, we said, generates an individual that has the same nuclear DNA as another individual. Just because they have the same genetic information, does it mean they're going to be exactly the same? No. Right. We talked to Michaela about that, and even as natural clones, she and her sister are very different, right? Because of the environment that they're exposed to. Okay, they can make different choices, they're exposed to different things, they have different likes, they have different looks and characteristics and features. So, same sort of thing. With embryonic twinning, you saw in the activity that basically the cells start to divide, they're pulled apart manually. So if it's an 8-cell, at the 8-cell stage, technically they could separate that and have 8 individuals, 8 clones formed that have the same genetic information. Okay? So they've done that kind of cloning for, we're going to watch a little video clip, we might not get to today, but about when they want to have dogs produced that have exactly the same kind of features, then they have done this in certain countries where, like for police dogs, they've cloned them in this manner in order to make them have the same features that are good for police dogs. Okay, so because they started from the same egg and sperm, the DNA is identical to each other. So they are like nature's identical twins, but they're made in a petri dish. So they would need a surrogate mother. Okay? Oh, at the bottom there it talks about fraternal twins. So what's the difference for fraternal twins? They start with 2 eggs and 2 different sperm. So because they have totally different genetic combination, they often will look very different. Might be a boy and a girl. Very different. Okay? Or they might have very different heights, very different hair color, eye color, everything. Okay? Sometimes they might look similar, but it depends on the combination of genes they get. They would just, basically, it's like they're sharing a room. They happen to be in the womb at the same time. So usually a female will only release 1 egg every month, every cycle, but the odd time, another one might come out, and if that one comes out, it can be fertilized by a sperm. Might not be the same donor sperm. So there have been cases where someone is carrying twins, but they had 2 different parents, 2 different fathers. We'll not get into that much. Okay? It's a rare occurrence, but it is possible. Yeah. But they can be twins because they're sharing the same, it's like they're in the same house, right? They're sharing the same mother. It's not a very pleasant thing to think about, but it is possible. Okay, so somatic cell nuclear transfer is the other type that you saw and how they cloned Dolly. Okay, basically this is a procedure that ends up getting an embryo. In this type of cloning, in reproductive cloning, the embryo is then put into a surrogate mother and allowed to develop, and then they're born as a clone. In other types of research, they might do the same kind of procedure just to get the embryo so they can harvest stem cells. Okay, so somatic cell nuclear transfer is really a procedure that can be common to both. In this case, we're looking at similar to Dolly, talking about a sheep. So just like in the mice, we have a tissue donor or a tissue cell donor and a donor that supplies the egg. I guess I can use a pen. This one, which one is it that you think that the end result is going to be a clone of? Okay, which donor? Right, okay, so because it's going to end up taking the nucleus from these cells, okay, so it's pulled the nucleus out into the pipette, and it's going to then insert it into this enucleated egg. The baby in the end will look like the somatic cell donor. Okay. There's no homework for tomorrow, but we have a lot to do tomorrow since there's no class on Thursday. And there'll be videos online, so yeah. The online assignment, Monday at midnight. Yeah, Monday at midnight. Yep. Okay. I have a few minutes, so I'm just going to keep going with this video so I can include it all as the cloning lesson. Okay, so Dolly is an example that was produced through somatic cell nuclear transfer. So she would have looked like the tissue cell donor that donated the nucleus from a somatic cell. This other donor, the one down at the bottom, is donating an egg cell, but the only purpose of having the egg cell is to put that other nucleus in. So they have to remove this egg nucleus, and then we call it an enucleated egg. Okay, and so then that gives a place for the nucleus from the somatic cell to be put into. It's given 1 to 6 hours to adjust and given some chemical to stimulate mitosis, and eventually we get an embryo. So in reproductive cloning, that embryo is implanted into a surrogate, okay, but the sheep that's going to be born isn't going to necessarily look like that surrogate mother unless that surrogate mother happens to be the one that donated the somatic cell in the first place, which doesn't have to be the case. Some pros and cons we talked about in the booklet. Hi. Thanks. Okay, I'll do it. Thank you. Okay, keeping on going. So, some of the pros: could help repopulate endangered populations, revive extinct species, reproduce deceased pets or loved ones, though remember it's not a carbon copy, and the ethics of that, well, kind of questionable. Some people actually want to do that though. So, could help infertile couples have children, produce animal models of disease that have more factors in common, may provide effective manner for producing individuals with special qualities. So we're going to look at, next day we can watch a video clip, and there's a link on the website if you want to watch it, about the cloned dogs. Okay, so basically seeing eye dogs, RCMP dogs, are now being cloned in some places to have better quality for their job in the world, or have if they're most efficient at that job, then sometimes they'll actually clone those animals. Again, ethics come into play, but it's happening. So, cons: very low success rate, expensive, higher rate of abnormal cell function in cloned individuals. Cloned individuals are not as healthy, or they don't live as long, and of course some ethical and legal considerations. But if you put that on your test or assignment, please make sure that you explain it a little further. Therapeutic cloning. The goal is not to create a cloned organism or a cloned human, for instance. Instead, it's to use the stem cells of the embryo. So basically, we get to the point of getting an embryo, and we harvest the stem cells in order to do some studies, to make tissues, or to treat diseases. So, how does it work? Starts exactly like what happened with the when we looked at the sheep model of somatic cell nuclear transfer. Okay, but at this point, when we get to the embryo stage, instead of being put into a surrogate like was in the sheep, the stem cells are actually cultured. And those cultured stem cells can be used in order to produce tissues, which might be used to build organs for transplant, or they might be used to study disease models, or other such things. Okay, so it takes about 5 days for the fertilized egg to develop to a point where they can harvest those stem cells. And again, there's still ethical considerations of whether that is proper to be doing. Pros: the hope is to use stem cells to act as replacement cells to treat diseases like heart disease, Alzheimer's, cancer, and other diseases. The hope is also that one day they can be used to create new organs for patients that are genetically perfect for their bodies. So if they use their own stem cells in order to produce the tissues, then they should be able to withstand a transplant because it's their own tissues entering their body. They wouldn't reject them like they would someone else's. So there's another little video clip that is linked from the website, but it shows that this is indeed happening, and it shows a successful trachea replacement. Sorry, someone else was talking to me at the same time. So have a look at that video. We'll try and watch it in class, but it's worth watching. So the third type is called recombinant DNA technology, or DNA cloning. It's also called gene cloning. So basically, we can take a specific gene or trait from one species and put it into another. For example, we have taken the gene for insulin and engineered bacteria to be little factories in order to produce it on high amounts. So we can produce it and follow, for instance, insulin. First they had to extract it, we said, from the pancreas of dogs and then cows, but they could only make so much at a time, and it wasn't human insulin. So this way they can use the gene for human insulin and make bacteria do all the work. They're also used in agriculture to give crops a certain resistance, so they might look better, or they last longer, or resist a certain parasite, so they'll withstand diseases too. Okay, so some of the pros may be used in gene therapy to treat certain genetic disorders by introducing a proper copy of the gene instead of a mutated one. They may someday be used to improve the taste, nutritional value, and resistance to parasites and increase yield of crops. They're already doing this for some crops, but they might be able to do it with more. Again, there's a lot of ethical concerns and concerns about possible side effects of doing that. We haven't studied the effects of making these alterations to the plants. Some of the cons, like I just said, introducing genetic new genes into an organism can cause defects or cause them to produce things that would be negative for the people that consume them or negative to the plant or animal themselves. And I think that's it. All right, that's it for the cloning section. Please make sure that you watch those other little videos and ask questions if you have any.